The availability of sex-specific genetic data, mtDNA for women, Y chromosomes for men, invites comparisons between male and female history. Mark Seelstadt, a graduate student of Cavalli Sforza's, chose to compare patterns of migration between the sexes. The logic is simple. Imagine a mutation that arises on a Y chromosome in Cape Town, South Africa. The speed with which it reaches, say, Cairo, is an index of rates of male migration. Similarly, the speed with which a Cape Town mutation in mtDNA reaches Cairo can be said to measure the rates of female migration. For good or ill, history has been much more the chronicle of men rather than women on the move. Typically, they were in search of plunder or empire. Think of Alexander the Great's march from Macedonia into the northern reaches of India, of the Vikings and their seaborne rampages from Scandinavia to Iceland and America beyond, or of Genghis Khan and his horsemen pouring across the steppe of Central Asia. But even without warfare as an excuse for travel, we still think of men as the more mobile members of human society. Men traditionally do the hunting, an activity that can often take them a long way from the hearth, whereas women in traditional hunter-gatherer societies stay close to home, gleaning food locally and raising the children. Therefore, Seelstadt had reason to expect that men would be our species' genetic prime movers. The data proved him startlingly wrong. Women, on average, are 8 times more mobile than men. In fact, counterintuitive though it may be, the pattern can be simply explained. Almost universally across all traditional societies, we humans engage in something anthropologists call patrilocality. When individuals from 2 different villages get married, the woman moves to the man's village, and not vice versa. Imagine that a woman from village A has married a man from village B, and she moves to B. They have a daughter and a son. The daughter marries a man from village C and moves to C. The son marries a woman from village D, and she moves to join him in B. Thus, the male line stays put in B, whereas the female line has moved in 2 generations from A to C via B. This process is carried out generation after generation, and, as a result, female migration proves extensive, but male migration does not. Men do indeed occasionally rush off to conquer distant lands, but these events are unimportant in the grand scheme of human migratory patterns. It's actually the step-by-step, village-to-village migration of women that has shaped human history, at least on the genetic level. Detailed regional studies of Y chromosome and mtDNA variation may also reveal something of the patterns of sexual relations and mating customs promoted in the course of colonization. In Iceland, for instance, which was uninhabited before the arrival of the Vikings, we find a marked asymmetry when we compare mtDNA and Y chromosomes. Most Ys are predictably Norse, but a large proportion of the mtDNA types are derived from Ireland. Apparently, the Norsemen colonizing Iceland took Irish women with them. Unfortunately, how the Irish women felt about this cannot be extracted from the mtDNA data. A recent study of Y chromosome and mtDNA variation in Colombia shows a similar effect. In most segments of society, Colombian Y chromosomes are Spanish Y chromosomes, a direct biological legacy of the European conquest of the Spanish Main. In fact, approximately 94% of the Y chromosomes studied have a European origin. Interestingly, however, the mitochondrial pattern is quite different. Modern Colombians have a range of Amerindian mtDNA types. The implication is clear. The invading Spaniards, who were men, took local women for their wives. The virtual absence of Amerindian Y chromosome types reveals the tragic story of colonial genocide. Indigenous men were eliminated, while local women were sexually assimilated by the conquistadors. Sometimes, however, enduring asymmetries are more a matter of cultural continuity than violent clash of cultures. The Parsis, a minority group in India, believe themselves to be descended from the Zoroastrians, an Indo-European Aryan people who fled religious persecution in Iran in the 7th century. Genetic analysis of modern Parsis indeed reveals that they have retained Iranian Y chromosomes, but their mtDNA tends to be of the Indian type. In this case, the asymmetry is maintained by tradition. To be accepted as a true Zoroastrian Parsi, one has to have a Zoroastrian Parsi father. Thus, membership in the Parsi community is paternally transmitted together with a Y chromosome. Here, genetics confirms the hold of tradition. Tradition has informed patterns of genetic variation among Jews as well. A recent study has shown that members of the priestly caste, the Kohanim, and their descendants, usually identifiable today by the surname Cohen, have a Y chromosome distinctive enough to set them apart from all other groups. Even the most obscure populations, those flung farthest by the Jewish diaspora, such as South Africa's Lemba, the Cohen Y has been preserved, almost like a sacred religious text. Its source is thought to be Aaron, according to Scripture, the founder of the Kohanim caste and the brother of Moses. It is certainly not impossible that the Kohanim Y chromosome sequence was indeed his, and that it has been passed down intact father to son in every generation since. Such have been the rigors of tradition over the course of Jewish history. Hammer and others have been able to use Y chromosomes to track the entire diaspora with interesting results. The Ashkenazim, for example, who have lived in Europe for the past 1,200 years, and now the United States and elsewhere, have nevertheless maintained the genetic indications of their Middle Eastern origins. In fact, molecular studies have made plain that the Jews, genetically at least, are virtually indistinguishable from all other Middle Eastern groups, including the Palestinians. So too is it written, Abraham, the great patriarch, is said to have had 2 sons by different women, Isaac, from whom the Jews are descended, and Ishmael, forefather of the Arabs. That such a deadly enmity should have arisen between the descendants of one man is an irony that grows only more bitter when genes seem to verify tradition's narrative.